Well, get started here. Yeah, so hello, thanks Jay for the introduction. I'm a research entomologist postdoc with the USDA ARS Pollinating Insects Research Unit, and I'm really grateful for the opportunity to participate in today's symposium and to present some of our ongoing work within this Benome 100 project on the evolutionary dynamics of bee genomes. Now, wild bees are critical to the health and productivity of the world's natural and agricultural ecosystems. There are around 20,000 species globally and at least 4,000 in the United States and Canada. And bees provide really essential pollination services. Although fewer than 20 species of bees are managed for crop pollination, there are remaining tens of thousands of wild, unmanaged bee species that likely contribute significantly to crop pollination. So both wild and managed bees are critical to the world's food systems. Bees are also an incredibly diverse insect group. Bees vary immensely in their size and behavior and life history traits, including their level of sociality, host plant specialization, and kleptoparasitism. Many bee populations are declining due to multiple stressors, including pathogens, habitat loss, and climate change, with several species currently listed as endangered or threatened under the U.S. Endangered Species Act. And so there's therefore an increased and really urgent interest in studying bee species to ensure a future with humans and the sustained health of our planet. Now, I probably don't have to spend much time convincing attendees of a biodiversity genomics conference why genomics is important. But for bees, genomic data provides insights into several key facets of bee biology, health, and conservation, starting with aiding the understanding of bee evolutionary and phylogenetic history. Genomic data can also be used to understand bee fitness, or the genetic underpinnings of decline, resilience, and health. Genomics is, of course, also critical for management and breeding, being useful for identifying management units, aiding the selection for resilience or traits of interest, and performing genetic rescue. Genomic data also provide reproducible, efficient, long-term data for monitoring and assessing species recovery. And genomic data can be used to identify distinct populations, identify inbreeding, and elucidate the demographic history of populations. And despite all of these advantages of genomic data for diverse bees, only a really small percentage of all bee species have publicly available genomes on NCBI. So this plot shows the number of genomes on NCBI by release year. The number of new bee genomes has been pretty low for many years, likely due in part to some of the technological challenges that are associated with obtaining high quality DNA from fresh, small specimens. More recently, there's been an upward trend in the number of genomes, in part due to improvements in technology and as well as global genome sequencing initiatives, including I5K and the Earth Biogenome projects. However, like I mentioned, there are over 20,000 bee species worldwide and around 4,000 species in the US, so we have some catching up to do. And that's where the Benome 100 project comes in. Formed in 2021, our mission is to sequence, assemble, and annotate high-quality reference genomes for at least 100 US bee pollinators, while also strengthening resources for the honeybee, which of course is a key pollinator and insect model. And there's a substantial focus on native bees of conservation, management, and agricultural importance. And more specifically, the Benome 100 project includes over 14 USDA labs and university collaborators. Benome 100 is organized into groups with project leads and participants. And then we have each of these subgroups that include topics such as bee diversity and conservation, data generation, processing and management, genome structure, sociality, and detoxification, among others. And the general idea is that once we generate the genomes, each group tackles a specific topic that excites them, which we hope will lead to advancing our understanding of and aiding the management of bees. And one of the key challenges for the project has been how to prioritize and acquire material for sequencing. Several members of the community have taken on this task, especially my co-author, Michael Brandstetter, who has helped determine which species to target for sequencing. And so, of course, a major consideration has been to acquire a taxonomically diverse set of bees in order to complement existing taxonomic representation of publicly available genomes. The team has also considered other features, including the agricultural importance, the conservation status, the life history diversity, and sociality. And of course, feasibility has been a major challenge, or has been a major consideration due to the challenge of getting fresh material. And so, like I mentioned, there's this challenge of collecting specimens because they need to be flash frozen in order to preserve high molecular weight DNA. And so the general procedure has been to net bees in the field, take voucher images while they're still alive, then freeze the specimens in liquid nitrogen and ship them off for sequencing. We also try to sample at least one voucher specimen that gets deposited in a major research collection. And unlike many other organismal groups, it's really important to make sure we carefully sex the specimens, given that females are diploid and males are haploid. And so when possible, we've tried to target haploid male specimens for sequencing. There have been many collaborators on the Beenomes 100 project who have very generously collected and submitted specimens for whole genome sequencing. And some of those individuals are shown here. So, okay, once we've collected our specimens in the field, the specimens have generally been sent to other ARS research units to perform DNA extraction and RNA extraction and to generate high molecular weight sequence libraries. We're targeting long read sequence data using PacBio, chromosomal structure information using Hi-C, and as much as possible, obtaining RNA-seq data from multiple tissues. Within the Logan unit, we're assembling genomes using scalable, reproducible workflows. And all of this has been a major team effort that's made use of resources and experience from many people, including those that are listed here. Once the genome is assembled and has undergone a plethora of quality assessment protocols, we submit the genomes to NCBI for public release. We annotate the genomes, and then we develop a variety of genomic projects using these note reference genomes. So where are we? The Beenomes 100 project currently actually has around 200 species in various stages of sequencing and assembly. So taxonomically, we've covered all US bee families, over half of the US genera. We also just recently reached a major milestone for the project where we uploaded the 100th bee genome to NCBI. So this is a phylogeny of ultra-conserved elements of bees, showing in blue text those species that have been added for the Beenomes 100 project, and in black text those that are already available on NCBI. And as you can see across the 6 families of bees in the US, we've added considerably to the taxonomic coverage, especially in those non-Apidae bee families. And as I mentioned when I introduced the project, there are many different subgroups of related applied and basic research questions. And so now that we have these 100 genomes available and have added significantly to the taxonomic coverage, we can really start delving into the science. And I'm going to focus the rest of my talk today on some exciting new analyses. I've started exploring the evolution of genome structure in bees, but I encourage you to stay tuned for developing research on all of these topics and more. So genome size evolution is a really fascinating trait that varies by orders of magnitude over viruses, archaea, bacteria, and eukaryotes. So this plot shows genome sizes for around 50,000 genomes available on NCBI. And even within groups such as plants, for example, the size of the genome can vary by fourfold. Genome size does not seem to be related to gene content nor to organism complexity, which is a phenomenon known as the C-value paradox. And repetitive element expansion is one important driver of genome size variation in eukaryotes and may play a role in determining genome structure and evolution. Repetitive elements can be categorized into subtypes, usually based on the mechanism of their duplication. And I'm not going to get too deep into the details of these specific categories today. But overall, repetitive elements can have negative effects if they transpose into a coding region of a functionally important gene, and for example, have been implicated in cancers and genetic diseases in humans and other organisms. However, they can also have positive effects by providing a mechanism for novel sequence variation or de novo exons in a gene. In insects, we know that repetitive element variation contributes to genome size. So this figure on the right is from a meta-analysis of 600 insect genomes in which Sproul et al. Found that larger assemblies tend to have a greater proportion of repeat content. And these authors found that insect genomes seem to contain virtually all known repetitive element classes, and they can range anywhere from 2% repetitive content to as much as 82% repetitive content. From the literature, we know that insect repetitive elements may be associated with ecological traits such as metamorphosis or kleptoparasitism. They can also be associated with the onset of insecticide resistance, as well as the evolution of climate adaptation. So they're really critical for multiple reasons in the evolution of insects. And so it can, of course, be hypothesized that repetitive elements play a potentially positive, negative, or neutral role in the evolution of bee genomes too. And so while this Sproul et al. Meta-analysis included some bees, there was a taxonomic bias towards species within the Apidae family. And so again, that's where the role of Benome 100 comes in. Sproul et al. Showed that long-read sequencing enables more complete assessment of repetitive elements. And we've added 100 new chromosome-level bee genomes on NCBI, which have a Hymenoptera busco completeness score of about 97%. This plot on the right shows the contributions of Benome 100 genomes and those chromosome-level long-read genomes bee genomes that were already on NCBI. And we can see that the taxonomic coverage of Benome 100 adds to many tribes of bees, especially Megachilidae, for example. So what do we see in terms of genome size variation? These violin plots show the range of genome sizes across 6 bee families from a total of 186 chromosome-level reference genomes. The orange dots are those genomes from Benome 100, and the purple triangles are those that were already available on NCBI. So first, we can see that bee genomes vary by a few orders of magnitude, with the smallest genome at 179 megabases and the largest genome at almost 3 gigabases in size. And some of these larger genomes are from species within the Apidae, many of which are orchid bees or longhorned bees. The plot also highlights, again, the contributions of Benome 100 towards increasing species representation within Megachilidae, and an overall shortage of species representation within Coletidae and Melitidae. Of course, these are some of the smaller bee families, but we do have additional reference genomes that we're processing. So hopefully we should be able to add to these numbers shortly. The number of chromosomes is also a fundamental way that we can describe an organism's genome. So again, bees show a wide range in the number of chromosomes, from the smallest at 3 to the largest at 36. The literature suggests that chromosome numbers follow a strong phylogenetic signal, and that changes in chromosome numbers can indicate major events such as polyploidy. But for now, I'll continue to focus on genome size variation, as this chromosomal number variation warrants further explanation at another time. So to further explore genome size variation in bees, we're implementing the repeat modeler, repeat masker workflow to characterize repetitive elements in all of our genomes. And we're still in the midst of running these analyses, so I'm showing you the results for 40 out of the 100 Benome species here on the left. Each of the colored dots correspond to a bee family, and we can see that there is a strong, highly significant correlation between genome size and repetitive element content, with some of our largest genomes having close to 90% repetitive element content. And it doesn't immediately appear as if there's a strong phylogenetic signal to this pattern among families, but obviously we need to perform more phylogenetically informed analyses next. This plot on the right is the same Benome data, except here I've added in the results from this Sproul et al. Paper, which includes the addition of 11 chromosome level genomes that were already on NCBI and underwent this same repeat masker workflow. And we see the same strong, highly significant correlation. So now that we've established this pattern between genome size and total proportion of the genome that consists of repetitive elements, we can use the output of repeat modeler to further classify the repetitive element content of these 51 genomes. So here on the left is this same phylogenetic tree of all NCBI and Benome 100B genomes, but I've just colored the species according to their family. And then this plot shows the assembly length, not for all of our genomes yet, but still for this subset of 51 genomes, because we're still processing the data. And so now we can look at the other variation of repetitive elements. So again, you can see that some of the largest genomes have the greatest percent of repetitive elements. You can also see some familial variation in terms of what percent of the genome can be categorized into lines, into long tandem repeats, or into DNA transposons. It's also really interesting to note how many of the repeats remain unclassified. So this is the total proportion of the repetitive content of each genome that's unclassified. And in these data, the genomes with the smallest proportion of unclassified repeats are all Apis species, such as the European honeybee, whereas many of the species that have a very high proportion of these unclassified repeats, we don't know yet what those repeats function as. And as I mentioned, we're in the midst of characterizing the repeats on the remainder of these bee genomes, so stay tuned for more analyses. One neat characteristic that we can look at with our current data is genome size variation in kleptoparasitic bees. Kleptoparasitic bees parasitize the nests of other bees, and this is an ecological trait that's evolved at least 20 times independently in bees. So these are each examples from 3 different bee families. It's hypothesized that there's a reduced genome size in parasites due to relaxed selection on metabolic and/or behavioral traits. So we might expect a reduced genome size and then perhaps reduced numbers of annotated functional genes. We find in our data that kleptoparasitic bees have significantly smaller genomes. So here I'm showing Violin plots of genome size variation for 28 kleptoparasitic bee genomes from independent lineages. Their mean genome size is 346 megabases, which is significantly smaller than that of the non-kleptoparasite bees at 489 megabases. We also see that kleptoparasitic bees have fewer repeats, so 29% repetitive elements in kleptoparasitic bees versus 47% non-kleptoparasitic bees. And this makes sense in our general relationship between genome size and repetitive element. And then we do also see that kleptoparasite bees tend to have around 2,000 fewer functional genes than non-kleptoparasite bees. So this is really exciting research that we're currently following up on. So I hope I've shown you that the Benome 100 project has broadened the range of measured repeat content beyond any previously reported values that we're aware of in bees. And we see genome size variation from 200 megabases to almost 3 gigabases. We see repeat content as high as 89% down to as low as 11%. And next, we can explore the evolution of repetitive elements in metabolism genes, for example, which is one mechanism that's previously identified for insecticide resistance. We can also explore whether certain gene families have expanded or contracted in specific groups of bees, such as generalists or specialists, which could improve our understanding of what makes certain bee species more amenable for a variety of pollination services or more resilient to pesticide exposure. And more broadly, I've shown you how the Benome 100 project may produce results that elucidate broad patterns of genome evolution across bees and provide an immense resource for further exploration by the research community and beyond. And so with that, I'd like to acknowledge the large group of collaborators of Benome 100, especially the Benome 100 leadership team, the sequencing and data processing team, plus all of the collaborators who are just poised to analyze all these genomes now that we have them ready. I'd also like to thank the symposium organizers for the opportunity to present today, as well as everyone for their attention. Thank you.
Great. Thank you so much, Ren. How about it? Let's do questions at the 40-minute mark, and we'll get Dong Ha to highlight the roles of NCBI and other groups, including EMBL, in making hay out of all these great genomes beyond the repeat structure. And Rena mentioned gene family expansion and contraction, and that's one of the immediate benefits of these projects, and that we need to have good gene predictions for. And I'm very happy to introduce Dr. Dong-Ha Oh from NCBI to explain exactly how they go about providing consistent and useful gene sets. So Dong-Ha, please take it away.
All right, yes, so let me share screen. All right, do you see the
Yeah,
right?
perfect.
Yeah, screen. Okay. All right. So yeah, hi all. This is Dong-Ha from NCBI RepSeq team, and I would like to first thank Dr. Jay Evans and the organizers for the opportunity to speak today. And also I would like to also thank Rena for a great introduction on the Benome 100 project and exciting analysis, especially on bee genomes being newly added. And following this, I will talk about a bit broadly on arthropod RepSeq genomes and tools available in NCBI to analyze them, and recent updates on NCBI author logs, especially for Hymenoptera. So Arthropoda Assembly Assessment Catalogs, or A3CAT, offers this overview of all GenBank arthropod genomes, updated, I think, monthly. And we see the four like large insect clades better represented here with several 2,000 species genome sequenced. And the quality of genome in terms of the assembly and 50, how fragmented the genomes are, and the BUSCO scores, how complete the genomes are kind of sort of all over here, but I guess that the more recent genomes will likely have a better quality. A bigger issue is that the vast majority of these genomes do not have a genome annotation. And from the NCBI datasets page, we can check the number of currently available genomes in any arthropod or any of these insect clades. As of a few days ago, we have 6,726 genomes from 3,819 taxa. Here, taxa mean mostly species, but they could also include subspecies. Of this, only 468 or less than 7% have genome annotation. Among them, 337 are annotated by NCBI RepSeq team, while the rest has annotation submitted by the user. And Hymenoptera has the second most genomes annotated. On RepSeq annotated 85 genomes from the same number of species. But considering that we have more than like several hundred species represented in NCBI, there are again a lot to catch up in terms of the genome annotation here. And if there is time and interest, I can later show a brief demo on how to obtain all of these numbers and information about these sets of genomes and download the genome data using the NCBI dataset page. Let me briefly introduce how RepSeq provides genome annotations. For eukaryote, we either import from curated resource for the model organisms, for example, Drosophila melanogaster, or run NCBI eukaryotic genome annotation pipeline, or eGAP, internally. And we are also developing a public version of eGAP called eGAP X. X stands for external. So that in the future, researchers can run the same genome annotation pipeline themselves. And our colleague Pooja is going to present eGAP X at the Genomics technology session tomorrow, so please join if you are interested. And back to RepSeq annotation, in principle, we annotate only one assembly submitted to GenBank per species. And the criteria to choose the assembly to annotate is the assembly quality and also the availability of RNA-seq evidence for the species. And vast majority of the new genome annotations are done per user request. And you can make a request with this button on the RepSeq eukaryotic genome annotation page in this URL. And once annotated, the RepSeq genome is incorporated to other NCBI products and comparative genomics resources. All genes in the RepSeq genome will be given a numerical gene ID and a dedicated gene page. And the genome and genes become downloadable through NCBI datasets. And again, tomorrow, our colleague Miriam will discuss NCBI datasets in the same genomics technology session. So please join if you are interested. And our RepSeq genome is also added to a genome browser called Genome Data Viewer, or GDV, where you can view gene models, RNA-seq tracks stored in SRA database, custom tracks, as well as alignments to other genomes if available. And Comparative Genome Viewer, or CGV, is an interactive viewer of pairwise genome alignments. And it has alignments between many Hymenoptera and RepSeq genomes compared to the honeybee genome available. So please check in this URL. And finally, gene ontology and orthologs are calculated for all genes in RepSeq genomes and added to the gene page real time. So for the rest of the presentation, I will focus on this NCBI orthologs. So one of the main purposes of NCBI orthologs is to name a gene by comparing it to a model organism reference. And to precisely name a gene, we only report one-to-one best pairs based on sequence similarity to both proteins and adjacent genomic sequences. And we also use the presence and absence of microsynteny of neighboring genes for tie breaking to identify the best pair when there are multiple homologous pairs available. And our approach scored one of the highest precision in Quest for orthologs, or QF evaluation internally. And since we call only one-to-one orthologs for vertebrates and arthropods, the recall was lower than other methods, but the precision was always among the highest. And NCBI author-logs are also scalable. Author-logs are added by pairwise comparison to a reference or an anchor at the time of the RefSeq annotation release. And it takes very low computational cost. Any number of genome pairs can be compared in parallel, and it will be finished often in less than half an hour. And we can expand to capture finer lineage-specific author-logs by adding new anchor species, as we will see in the next few slides. And let's look into author-log anchors a bit more. This is a tree of all RefSeq metazoa genomes based on Busco sequences. Metazoa is animals. And on the vertebrate side, we have mammals, sauria, and fish. And zebrafish is the author-log anchor for all of the fishes here. And human here is the anchor, author-log anchor for the rest of the vertebrates. On the invertebrate side, RefSeq has a better representation of arthropod. These are all arthropods in NCBI, RefSeq. And especially insects, we can see the major clade of insect studied as disease vectors, pests, and pollinators, of course, and model systems for various other research topics. For this broad space of arthropod, we have only one model species as an anchor, Drosophila melanogaster, or fruit fly. Now let's see some example author-logs calculate for first Diptera in this clade with the fruit fly as the anchor. So here a dozen Diptera species ordered roughly by the distance from the anchor Drosophila melanogaster or fruit fly. And the closest Drosophila species reports more than 90% of the entire protein coding genes as author-logs. But even within Drosophila clade, the number of author-logs begin to decay as we go further from the anchor. And the number of author-logs are lower for other flies and down to several thousand in mosquitoes. And here there is an N. N is the number of neighboring genes showing synteny, and which is synteny means the conservation in gene positions and orders. And since we search 10 genes upstream and 10 genes downstream for synteny, the maximum N is 20. So similar to the number of author-logs here, the N is close to 20 for the closest species, Drosophila simulans in this case, and become lower for more distant species. And how about calculating author-logs between these Hymenoptera in different order with fruit fly as the author-log anchor? We see that N is now very low, meaning almost no synteny signal. This indicates that a fruit fly is quite distant from all of these bees, hornets, ants, and wasps. And however, the number of author-logs are more or less stable. These several thousand may represent author-logs deeply conserved across different insect orders. Next, we try the honeybee as the new anchor. Honeybee located here. For all Hymenoptera RefSeq, honeybee can be considered a semi-model species supported by a strong research community. So we choose it as a new anchor to cover the Hymenoptera clade. And here I am showing the number of author-logs anchored to fruit fly and anchored to honeybee side by side. With the honeybee as the anchor, we see a quite substantial increase in the number of author-logs being reported. And N increased a lot also as well. And especially for the bees, the closer to the anchor, honeybee anchor. And this will help improving the resolution of the author-logs. Even for more distant wasps here, we see an increase in N. And also like about 20% more protein coding genes reported as author-log compared to when fruit fly was used as the anchor. In all, we are capturing additional 18 to 36% of the total protein coding genes as also log, which are likely specific to Hymenoptera. So the honeybee anchor benefits 84 rep-seq Hymenopterans, and the number is increasing. And we can also use log data to improve the anchor honeybee gene annotation as well by feedback mechanisms. For example, if most of these 84 Hymenopterans have an logs in more distant fruit fly, but not in honeybee, then the problem might be on the honeybee gene annotation actually. So in this way, we have already identified cases where we can do curation to improve honeybee gene models. So here is a summary of numbers. In addition to the 2.11 million logs anchored to Drosophila melanogaster gene, just earlier this month, we begin to report almost 250,000 more Hymenoptera specific log anchored to honeybee genes. And we are considering additional log anchors for other insect clades as well. Now, how we can access the NCBI logs? First, we can start from NCBI gene page. So each NCBI gene is given a numerical gene ID and its own gene page that looks like this. Here, you can click the log link at the summary section or go to the general gene information section, which include links to both NCBI logs and also on external log resources called OSODB when it is available. The log link from gene page brings you to this dataset page showing the log set. Here, the gene named Delta has logs found in 277 species across all arthropods. Some log sets are found only among Hymenoptera. For example, Hymenopteacin, an antibacterial peptide, is found among a subset of 50 Hymenopterans only. And some logs are really restricted to only the genus Apis. Apis is a peptide building block of royal jelly, and it is found only among honeybees. And here you can select all of these logs by clicking here, and either to download the sequences, or you can create a quick protein sequence alignment, as in this example. If you are familiar with NCBI datasets, you can use its command line interface to download logs for multiple genes from a set of taxonomy ranges. I'm not going to details here, so please visit the how-to documents for details. The URL is a little bit long, but you can also Google simply NCBI datasets logs to find this page. Finally, all logs we report can be downloaded from NCBI FTP. The gene_logs.gg file here contains a simple tab delimited table of taxonomy IDs and gene IDs of the anchor and log pair. So future work includes the expansion of anchors as mentioned earlier, and more importantly, we plan to release this NCBI logs method as a public pipeline, either as a part of the eGapX or as an independent pipeline, so that the user can generate logs for clades and reference anchors of their choice to serve their own research needs. So with this, researchers can calculate logs for any pair of rep-seq genomes based on gene locus, and multiple comparison can be made in parallel and in scale with relatively light computational footprint. And as a part of the annotation process, we also calculate Panther protein family for all rep-seq proteins, which can be a resource to report paralogs if needed. And the most important message of this entire presentation is that we would like to hear user stories and feedback. How do you use logs or paralogs in your research, or any pictures or data you would like to have? And you can see this feedback button on almost every NCBI page. So please use this button, or you can email cgr@nlm.nih.gov. Here, CGR stands for Comparative Genomics Resources, an NIH project that covers all resources mentioned in this presentation. So this presentation was on behalf of the LabSig eukaryote dev team and prepared with help from people in and out of NCBI. And thank you, and I appreciate any comments or questions.
Great, thanks so much, Dongha. I would like to get back to Rana and the Benome. We have time is short. We have about 8 minutes on Benome, and then we'll shift to another more mature pest genome, insect genomes project. But Dongha, I just wanted to highlight one of the chat questions from Mark Blackster, and I've thought about this too. Is there are other third-party annotations for genomes? I know that INSDC is sort of set up to make genome data interchangeable and sort of easily merged. Are there barriers that could be relaxed a bit to get to get out other annotations for genomes into the both NCBI and then in the converse into the European and Japanese databases for genomes? Is it such that genomes generated in one place are hard to integrate with genomes from another, or sorry, gene sets in one place are hard to integrate, and is that something we could help spur somehow?
Yeah, so my understanding is that like the eGap X is actually an attempt to help the researchers to make it easy to submit the gene annotation. And like genomes that submitted to any of these any of these three database will be shared, but I'm not sure about the gene models. So yeah, for that I will have to ask my team members. So I will note this question and get back to you. But the eGap X might be one of the like answer we are trying to provide. Yeah.
Yeah, thanks. Yeah, and Mark noted I did speak because ensemble annotations, of course, are from an equivalent
Yes, yeah,
body.
ensemble is yeah.
Yeah, so great. But yeah, we're as consumers of genomes and gene sets, we're just happy that you all work well together. So oh, there's one question from Anna Ramucci. Let's cover that really quickly. Can you see that, Dongha?
Is there a way to track genome use or downloads on NCBI? It would be useful to be able to look up a genome and then see that it's always associated with 3 publication or has been downloaded 100 times, tracking the use of the. So use of the genome. So if you go to the genome database data sets, then genomes are actually ordered, and the order seems to be a little bit like random. But actually, they are ordered in the number of bio project that is providing the data for that species. So, like Drosophila melanogaster will be like studied by the multiple groups. So they will be like comes up earlier than other genomes. And so if you see the genomes in the earlier part of the list, then there is a chance that they are the one that is studied more. But for the download and publication, this kind of thing, I think is kind of hard to track. And also this order of the genome is also like once you go below certain level, then many of them will have just a few bio project associated with that species, and the resolution is not that great. So there is a limitation. But this is a very good thing that I can also discuss with our team members. I hope we can have the chat.
Yeah. Great, thank you for that. Okay, now let's go back. Does anyone have questions? And I think you can actually unmute should you wish and talk or chat. Any questions for Rana about the Benome project?
Well, this is Clement Kent. The question I had to ask is, what speed do you see your added Hymenopteran genomes getting into RefSeq?
That's a great question. And we've been in touch with folks at NCBI on two things. First is we are hoping to use the eGAPX pipeline to annotate all of our 100 genomes. We have RNA-seq data for probably 75% of those genomes, so that will greatly add to our ability to have good gene annotation sets for them. And then as Dong Ha mentioned, the idea of the eGAPX is to then be able to integrate that annotation into NCBI and the RefSeq database. And then we're also planning to have around 20 of our genomes annotated by NCBI, as well as with the eGAPX workflow, to further kind of test how well those two different approaches to gene annotation work. And so we are, now that the genomes are ready, we have most of the RNA-seq data, we're really focusing on the gene annotation next.
Cool. Thanks, Rana. There's a quick question from a bee fan from Stephania, which is that wondering if the kleptoparasite patterns that you described for genome size, if they could be explained by particular families, or if there are enough independent contrasts in different families that it could not, it's not a phylogenetically leggy sort of result.
Yeah, that's an excellent question. That is a very new result. So I need to spend more time looking into those results further. The results I showed you did come from 3 different bee families. So we do have genomes already for at least, or kleptoparasitic and non-kleptoparasitic for 3 different bee families. But that's kind of all I know right now, and I definitely we need to look into that further. That's a great question. Thank you. Can I go ahead and answer those other questions, Jay?
Oh, of course. Yeah, go for it.
Yeah. So the Beenomes, for individuals, for each species, they are mostly individual. When possible, we try to use the same specimen for both Hi-Fi and Hi-C and RNA-seq data. But of course, that depends a bit on the size of the bee. So for some of our really small bees, for example, some of the Andrenidae are really small. There are a couple of genomes where we had to pool like 2 males to get enough DNA to be able to do the long read sequencing. But for the most part, they're from the same individual. Oh, and then in a lot of cases, the RNA-seq data is from the same individual. But just like the DNA, it kind of depends on the size of the bee and how the library prep itself went. And so I don't know the exact proportion, but we have a mix of RNA-seq data that's from same exact bee or other individuals, but the same species, and usually the same exact sampling event. So trying to get the same location information for each genome. I'll just keep answering if that's fine, Jay.
Yeah, of course, go for it.
Yeah, I think we're using low input protocols for PacBio, the Hilo team. They're the ones who've been doing the majority of the library preparation and sequencing. And I know that for that they're exploring some of these ultra low input protocols because some of these bees are really small and we don't have that many specimens. And so they're working on on getting those protocols running so that we can use those ultra low input on some of the bee genomes. We do screen our genomes for any non-arthropod DNA before we upload it to NCBI. And of course, that's the contamination screen is also part of the NCBI workflow for quality check of all genomes that are uploaded. But we use the blob tools workflow within our own pipeline to isolate all of the non-arthropod DNA that will then go to interested subgroups who are interested in microbiome data and other sorts of contaminants to our bee species. And then hopefully, as good as our resources are, our databases are, our pure bee genome is what's uploaded to NCBI.
And just again, if there are people on this Zoom, there is a rabid group looking at microbes, and but also if you have a pet group of microbes, get in touch. We're also exploiting the RNA data for some RNA viruses that are important for bees. And so yeah, it's so far I will say it's very spotty. Some samples have a lot, some have almost none in terms of hitchhikers, but as to be expected from one bee at a time. But hey, I think we should shift. They're great questions, but just to give Dr. Anna Childers is on, maybe a few minutes to highlight a sort of parallel, but a parallel project tackling some less beloved insects that are often crop pests and things like that. And but we're kind of doing this in parallel to the extent that the technologies are quite the same and some of the people are quite the same. So Anna, why don't you give like 4 minutes on that, and then we'll shift to Rich and the beetle world.
Sure. Yeah, I just want to say, so the Ag 100 Pest project is the one that we started off with at USDA, and this is really where we've tried to tackle a lot of these challenges. And then thankfully, based on the success of this project, the Benome project was given congressional funding and was really able to take off from the foundations that we generated. So when we talk about the Ag 100 Pest, it's really a project that is really thanks to the folks on this slide and all the work that we are doing across the entire agency. You know, I've kind of highlighted where some of the leads are located, but we have species coming in from across the country and collaborators across the country working with us on this project. And one of the big things that we've really tried to do over the last 5 or so years is to really improve our pipeline here, improve the quality of what we're generating, decrease the time of how long it's taking, and everything else. Our goal is to always get the long-read sequencing from a single specimen. Sometimes we have to do the high-C from a different specimen, like Brenna was saying, but we really want to base that sequencing on the long-read sequencing on a single specimen as much as possible. And so this pipeline has been tuned and retuned over time. We started back in the days of CCS sequencing, and obviously we're using hi-fi sequencing now. So this is a big change. One of the things I want to emphasize is that there's a lot of challenges with sequencing arthropods. Anybody who has done it know that some are just very difficult. But there's a huge variation in genome size. So we've done several Orthoptera now, some of them with genomes up to almost 9 gigs, right? And so thankfully with a locust, you have quite a bit of material to work with. The bigger challenges in arthropods are really things like our ticks that have maybe a 5 gig genome, but are very tiny physically. So some of the big improvements that we've been able to make over the years have been working with PacBio on lower input methods and things to improve the sequencing options and the ability to sequence things with very small genome sizes, and really trying to tackle that biggest challenge of very small body size but large genome. And so when we think about this project also, we can really think about diversity. We're handling a lot of diversity in terms of the agricultural stakeholder groups that we're trying to help out, from various crops and plants that need additional resources to our bees, the bee pests, and animal pests and stored product pests. But the other way to think about diversity is the diversity of species that we're sequencing. And so this project has really been able to help ensure that there are reference genomes available for many additional families. And then when we talk about the comparative genomics, we're also trying to make sure that there's much deeper representation within some of these families for comparative work. And obviously goat is a great place to really dig into that further. One thing that makes our project unique, and the Benome is also doing this as well, is that the I5K workspace at the National Agricultural Library, which is part of the USDA, is taking these species and making them available, is adding functional annotations, is making genome browsers and the ability to do manual annotation and other sort of things available for our community, which is a real added bonus. And so with that, these are some of the iconic species that we have sequenced, and I'll just leave it at that and see if there's any questions. Be happy to answer them, but I think like we said, or like Rena and others have said, a lot of what's getting there's a lot of similarities here with the Benome project, and in terms of the not just the people, right, but the actual infrastructure behind it, the methodologies that we're using and things like that.
Cool. Thanks for talking so quickly, Anna. Any questions for Anna? I'll look in the chat for you.
And I think I put it in the chat, but Clement, to address your question about the speed at which these genomes are becoming available, that's something that has really varied over the years. And with time, obviously, there's a lot of people dumping a lot of genomes onto NCBI right now, and I'm sure that they can speak to the strain of that. It's and certain genomes just seem to be a little bit more difficult to get through, or they just seem to take a little bit longer. And I'm not exactly sure why, but I think we should have at least about 86 that are publicly available of ours. And I know Rena has done an amazing job getting those bees out. So these, again, if they're not publicly available, it's not that we aren't trying to release things. That's actually part of the USDA's policy of open data, is to get these things out as quickly as we can. It's just some of it is just the challenge of getting some of that final public release done at NCBI.